Today's objectives, we're going to describe the organelles, we're going to finish the non-membranous organelles and the membranous organelles and their specific function. We're going to explain the functions of the cell nucleus. We're going to summarize the process of protein synthesis. We're going to describe the stages of the life cycle of the cell, including mitosis, interphase, cytokinesis. We're going to discuss the relationship between cell division and cancer, and we're going to define differentiation and explain its importance. The last non-membranous organelles include the centrioles, and they move the DNA strands in cell division, the cilia, and they move substances across the surface of the cell, and the flagella look like a long cilia but are used to move the cell through its environment. The ribosomes, and there's two major types, free ribosomes and fixed ribosomes. The ribosomes manufacture protein. The free ribosomes are spread throughout the cytosol, and the fixed ribosomes are attached to the ER. The proteasomes are organelles that contain protein-breaking proteolytic enzymes, or proteases. Now we're going to begin the membrane-bound organelles, or the membranous organelles. And the first one is the ER, endoplasmic reticulum. It has four major functions. It synthesizes proteins, carbohydrates, and lipids. It stores materials. It transports those materials through the cell. It's involved in detoxification of drugs or toxins. There are two types of ER. There's a smooth ER, SER. It has no ribosomes, and it's where the lipids and carbohydrates are made. And then the rough ER, the RER, and it has the fixed ribosomes, and this is where the proteins are made. Here's an image of the ER, and you can see the smooth and rough ER. Next, we have the Golgi apparatus, and it's made of flattened membranous discs called cisternae. It has three major functions. It modifies and packages secretions, it renews or modifies the plasma membrane, and it packages enzymes. Here is an image of the Golgi apparatus and it releasing its secretory vesicle. Next is the lysosomes, and they're produced by the Golgi apparatus and they contain digestive enzymes. They help with cleanup and recycling of materials within the cell. They help with autoimmunity and they can trigger autolysis. Peroxisomes are next, and they're formed from existing peroxisomes. They're enzymes that break down fatty acids, and their byproduct is hydrogen peroxide. Hydrogen peroxide is a free radical that is highly reactive and can be destructive to cells. Then we go on to the mitochondria. The mitochondria is formed of a double membrane. The outer membrane surrounds the organelle. The inner is folded to form the cristae. The cristae increase the surface area exposed to the fluid matrix. It's the site of ATP production through aerobic metabolism or cellular respiration. This is an image of the mitochondria. The nucleus is the largest organelle in the cell, and it's the control center of the cell. It dictates the cell function and structure by controlling protein synthesis. The nuclear envelope is a double membrane that surrounds the nucleus. It has nuclear pores that allow some movement of substances in and out of the nucleus. Next, we have the nucleoli, and that's found in most nucleus, and that's where the ribosomal RNA is made. The chromosomes are found in the nucleus, and they store instructions for the protein synthesis. Chromatin is loosely coiled DNA, and it's found in cells that aren't dividing. Here's an image of the nucleus. This shows you the DNA, the chromosomes, and the nucleus. Remember that the DNA stores the genetic information that dictates an organism's inherited traits. The genetic code is arranged in triplets. Know that for the test. Genes are the functional units of heredity. Next is protein synthesis. The DNA controls protein synthesis. This is when protein is actually made by the cell. There are two stages, transcription and translation. Next, we're going to go into the first stage, transcription. Transcription, it deals with the messenger RNA or the mRNA. This is to get the DNA blueprint for protein synthesis from the nucleus out to the cytosol. The RNA polymerase binds to the promoter gene and unzips the DNA. The new RNA codon triplets are formed. At the DNA stop signal, the mRNA detaches and the unzipped DNA reattaches. At this point, the mRNA will leave the nucleus and go to the ribosome. This shows you the illustration of what we've just talked about. Next is translation. That uses the new mRNA codons to signal the assembly of the specific amino acid in series. The mRNA leaves the nucleus and goes to the ribosome in the cytoplasm. The tRNA, or transfer RNA, delivers the amino acids to the ribosome. Each tRNA has a complement to the codon called an anticodon. This illustration shows how the tRNA brings the amino acids in sequence and makes the new protein. This table shows you the DNA triplet code, the mRNA codon, the tRNA anticodon, and then the amino acid that is produced. Next are the stages of a cell's life cycle. Cell division is the growing of a new somatic cells and replacing the old. Mitosis is the DNA replication of the genetic material in the nucleus, or the duplication of the nucleus. Meiosis is the production of sex cells, the sperm and the ova, and then apoptosis is the genetically controlled cell death for cells that do not divide. Interphase is the time a cell spends performing its functions and preparing for mitosis. It has G1, that's the phase when the cell duplicates organelles and adds cytosol, S when the DNA is replicated in the nucleus, and G when the centrioles are replicated. This shows you interphase, G0, G1, S, G2, then mitosis and cytokinesis. There are 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. In prophase, the DNA has already been replicated, the nuclear envelope begins to disappear, and the DNA, or the chromosomes, become tightly coiled and are visible. In metaphase, the chromatids move to the central zone called the metaphase plate, and the chromatids line up parallel to the plate. In anaphase, the centromere of each chromatid spins, creating a daughter chromosome. The daughter chromosomes are pulled apart and moved towards the centriole. In telophase, the nuclear membranes reform, the DNA uncoils, and the cells are preparing to enter interphase again, and we have 2 complete nuclei. In cytokinesis, cytoplasmic division takes place and it completes the formation of 2 daughter cells. It usually begins in late anaphase and continues throughout telophase. Here we see the illustration of interphase, prophase, metaphase, anaphase, telophase, cytokinesis, and interphase again. Cancer is a result of abnormal cell growth and division. Benign tumors are usually encapsulated and rarely life-threatening. Malignant tumors spread from the original tissue capsule through a process called invasion. Having spread to other tissues and organs, the tumor has metastasized. Cancer is the result of very active malignant cells, and it stimulates additional blood vessel growth, and it also triggers positive feedback mechanism of further growth and metastasis. All body cells or somatic cells in the body have the same number of chromosomes and genes, yet they develop to form a wide variety of cell types. Differentiation occurs when specific genes are turned off, leaving the cell with limited capabilities. A collection of cells with specific functions is called a tissue.